we're at the point, I think, of having an open discussion and questions for specific speakers. I want to at least start with some of the questions that have popped up so far during the talks. The first from the Google documents, Natalie Lynn, this is from Megan. She asked, for the zebrafish studies, how long are these studies done to come up with these conclusions? It seems that the life cycle of zebrafish is very fast, which allows for quicker results. Is that accurate?
Hey, thanks for the question. So many of the results we're getting, we can get within 5 days post-fertilization. So, I mean, we can, do our initial experiment, do our pooled CRISPR knockout, and have a morphometric phenotype within 5 days. And so it's a really nice way to be able to quickly kind of screen through a wide number of genes in a fast manner.
Can you also elaborate, I think, for the group who might not know, like how many zebrafish do you get? How given week? How often do you breed? How big is a colony? Something like that, compared to say mouse or the time scale and the scale of the projects.
Sure, that's great. So, so at Davis, we have a relatively small facility. We have 2 double racks, and so I think it can have something like 7,000 adult fish, which isn't a whole lot. It may sound like a lot, but it's not a whole lot, but you can fit them in a small space. And so for every kind of given breeding pair, you can produce a couple hundred embryos per breeding pair. And so you can set up, in parallel, many different breeding pairs. And so you can produce thousands of embryos per day. And because many of the experiments we're doing are within wild type fish, and then we're introducing the CRISPR and doing this pooled knockout, we don't have to have a lot of space to carry a lot of these different stable lines, which would take up a lot more space. We can do a lot of initial screening in these, starting with the wild type, and then testing these mosaic fish. And so, and if we were to be able to also couple it with the mic drop method, which I mentioned, which you can actually in a single injection inject up to 100 different genes. In a single injection session, you could imagine being able to screen hundreds of genes across thousands of embryos within a matter of kind of a month, being able to have at least a first pass on how these genes would implicate and change morphometric traits. So the scale of it is just really great. I think zebrafish, they are a vertebrate system. They have a complex brain. They undergo development. And so I think it's a great system for testing at scale. And also with the drug, what Scott mentioned with the drug rescue, it's also really useful. So.
I guess as a quick follow-up while we have you, Eric Marsh asked, do you ever let any of the fish that you manipulate grow to adults, and do the adults have phenotypes? I can comment after you.
Yeah, maybe. Yeah. I mean, so to be honest, the majority of what we do is within the embryo, but we do let them grow to adults because we have stable lines, and so our stable lines grow up. And we do see that some of our adults in the tank are suffering from behavioral deficits, but we haven't formalized that for our fish. But it's because we're characterizing kind of many genes. But if you were to go deep into a single gene, I think that this is something, for instance, Scott, you're probably doing.
Yeah, I would say, we certainly grow our CRISPR lines to adulthood. In fact, our project is based on using F3 generation, so they're a little more stable and to eliminate any side effects. Eric, I would say my experience is there's really no good reason to do an adult zebrafish because you end up with 1 or 2 fish, and the time frame is the same as a mouse, and so we can start to do the types of stuff that Min-Chan does in a mouse without the zebrafish. And you'd eliminate a little bit of the bias against zebrafish if you publish an adult mouse versus an adult fish. So I don't really see a big advantage. The big advantage is the numbers and the things you can do with larvae. Once you get to raising a fish for 3 to 6 months to do an experiment that might be less popular, is the best way to put it. I don't see where the fish remain an advantage over using a mouse or an organoid or something that you are already looking at for a long period of time. Yeah, but they certainly have adult phenotypes, and lots of labs publish on adults. I just don't think there's much advantage to it.
Yeah, I totally get that and understand, and I appreciate that. I just was like the developmental aspect of it from larval to adult, what changes developmentally, and can we learn something in looking at that could be applicable to all vertebrates and to be, to give us more insight into the disease process. That aspect of it, people don't use because it is time consuming and more problematic. Totally agree. And its power is what you're saying, but just that aspect people don't talk about much. I was just wondering if, more comments on that.
Well, Megan can add more because she's more of a developmental biologist, but their brains are very different. So you've run into, well, I think Megan just explained how their brain is different. It'll make more sense to the clinicians.
Yeah, I mean, so fish don't have a neocortex, and so I think like many of the early neurodevelopmental traits we can say are conserved in terms of like neural migration, synaptogenesis. But I think as we move later, I think we're kind of going farther away from humans as well. And so, I mean, I do think that there's utility in it. And, I myself haven't done any work past 5 days post-fertilization personally, mostly because I'm really trying to do this at scale. And so, I think that there is a possibility to do that, and especially with LGS, with the kind of progression into later stages. But this is not something that I can really speak to very well, to be honest. Really focus on neurodevelopment. So,
and I think the developmental question, even earlier, I think Anne put something in about looking, or Kelly, different stages of development and progression of, say, like, electrical activity and how that would progress in a patient. For fish, at least, my experience is, one day is like a long time. It's almost like a month, roughly, it seems. And I learned this by trying to kindle them in the early years. And so no matter how many different ways we would zap the fish, and get them to seize, we'd come back the next day and they'd be perfectly fine. So I think they're very rapidly developing. And so you might not see that progression, and also lacking all the structures you might need to get a more complex EEG event that you see in humans has always been the problem I have with animal models in general. What are their limits on the type? Like hypsarrhythmia always comes to mind for me. In animal models, you might not have the circuitry required to generate hypsarrhythmia. The organoids, I think, and Allison maybe hopefully can comment, I think because he can keep them for so long and monitor the same ones and watch the progression like he's done of oscillations, I see hope there to kind of see the progression that might mimic human electrical patterns, but maybe Allison can comment more.
Yeah, you're absolutely right, Scott. That's one of the advantages. But the organoid model, it doesn't come pre-made, right? I mean, that's the difference from the other models. I mean, if you take any animal model, they will become the animal. The organoid model will become what you want. And here's the tricky part, because, I mean, we don't know much about the disease to know what kind of pieces we need in the organoid. I'll give you the example of the CDKL5. We have been working with that syndrome for a while. So yeah, we got the difference in oscillations. It's not quite yet the seizure activity. But now, what if we add the thalamus? Now we have the cortex thalamic talking to each other. And our hope is that now we're going to see it. And we are using that not only to create a better model, but also to understand where the seizure starts. And that would inform, for example, where the gene therapy should target. Should we target the cortex or should we target the thalamus? So the model is like, I like to think about a Lego thing that you're going to be building and creating as you go along to understand the disease and improve the model as you go with it. And in the meantime, I mean, some of the insights that you can take could be an opportunity for therapies or clinical applications down the road. But yeah, I mean, it doesn't come pre-made. You have to know exactly what you want and kind of building that.
No, that's a great point. And I think that also ties in with Minchan's work that might hint at which of the components you want to add. I mean, inhibition versus excitation broadly, like he's shown for specific phenotypes. But then you get into, like I said, either excitatory cells from different brain regions have different purposes, interneurons, you get into the complexity of different types of interneurons going from, 3 subclasses to, 50 if you look at the Allen Brain Institute data, and adding that. So that's how I kind of wonder, I guess, just my personal question, do you kind of follow that? Because I kind of do that for fish, where I try to follow what the clinical features are as they emerge to see if I can add any of them to my fish. Obviously, the fish is a little bit more pre-made and we have to alter it in different ways, but it strikes me the organoids allow you to kind of monitor those clinical features. And like I said, add them, could you add a dysplasia to, a different part of the circuit or whatever, things like that, like I think Lego's a great, Lego system's a great idea, analogy, but.
Yeah. The effect in the cortical dysplasia, which is kind of an idiopathic condition. Yeah. And we saw, things like the dysplasia in the cortex plate, the balloon cells, the multinucleate neurons. The fact that we see that, I think it's very positive. It's kind of mimicking what we see in the tissue of the brains. So that's pretty good, actually. I to be honest, I was not expecting to see that.
Yeah, me too. I want to, I guess, echo something that Tracy and I talk about a lot. We used to work a lot on focal cortical dysplasia here in rodents. So we never really got animal models that had, really convincing balloon cells. And we're always, and Tracy knows this, I always got their feedback like, well, it's just not possible, instead of the flip side is like, it is possible, we just were using the wrong system. I think Tracy likes the idea as well. It's like, we as scientists or clinician scientists have to stop looking at some of the experimental techniques that are out there and just dismiss them as not being useful, when it might just be that we just maybe are at the wrong stage of the development of that process or technique. And these things are possible. The open-mindedness, I think, Tracy and I kind of share in this of to pontificate a little bit and the AS community, the epilepsy community, to not continually say something's useless or not possible, and maybe start to take the idea that it is possible, we just haven't figured out how yet. So, sorry, Tracy, on that slight side rant. Well, I'll go back to the questions. So from Jenny, and then we'll get to the bigger discussion, I think, that Anne and Kelly and others and Eric have started to put in here, which is really critical. So do you want to ask Jenny, or do you want me to read it? I see you're live.
I can ask.
Yeah, better to do it live.
Hi, everyone. Great session. So Scott, I remember in your introduction that you mentioned that some of your zebrafish lines, they showed some interesting responses to the different drugs, the different anti-seizure medications that weren't necessarily being seen in some of the mouse models with the same genetic mutations. So I was just trying to connect that to Megan's talk, where she mentioned that in her genetic model with the partial duplication, and that perhaps that might be more, have the ability to mimic some of these human-specific phenotypes or traits. And I'm wondering, do you think that could be an explanation why there could be some of these responses observed in zebrafish and not in mouse mutants?
I would say on the side of the drugs themselves and their efficacy, and I know Jennifer's on here, and I never mean to dismiss Jennifer's work. It's just, in that case, an example. I think the difference my explanation for the difference is that in zebrafish, I'm able to study spontaneous seizures, right? Electrically. I don't have to manipulate it like the AED programs work. It's not a seizure susceptibility issues. The fish have seizures, and I look for drugs that stop seizures. So I think that allows them to be predictive of AED's efficacy in a way that mice aren't. I think mice would be if you did the same experiment. It's just, as Jennifer would say, logistically impossible to screen, 3,000 drugs in a mouse using spontaneous, video EEG monitoring. So I think when Jennifer as she gets more data, and again, it's not a fair comparison. Obviously I slant it when I give my talk because, she's only able to test a few drugs at a given concentration. And the other thing is, like, I can test many concentrations and exposure time. So there's a whole bunch of issues of why maybe the drugs didn't work in the mice that would require just more experiments. But the problem I see is those experiments could take decades to actually get all that data. But I think it would match up eventually. I just don't think it's easy. Maybe Jennifer thinks, can comment.
Yeah, I can elaborate on that a little bit. I think that we published our screen, and we were quite honest that if you only sample 60 hours in a mouse at a given concentration, you may not see an effect. And it's not that you don't see an effect of the drug, it's that the seizure frequency is too low to see an effect. So, since then, what we do is we look at the animals for a week or two, and we get a much better seizure frequency, and we can actually now see drug effects. And they are predictive, although I will say that some of the serotonin modulators are a little bit trickier. That's where maybe the fish are coming out ahead. And we haven't spent a lot of time on those because the fish have done a good job and jumped to humans. So there doesn't really need to be a sidetrack into mice at that point.
Thanks, Jennifer. And Megan, maybe comment on that if you want me to know more about the genetics.
Sure, yeah. So I guess just one thing to just piggyback on the statement, I don't think that there's one perfect method. And I think with my use of the zebrafish and others, we really are thinking about this as using multiple different approaches. So, starting with fish, testing many, many genes in parallel, and then going into a mouse or an organoid model that maybe is more expensive or harder to do on at scale. And so that's one thing. In terms of the kind of duplication landscape. So I was actually testing a human-specific duplication. So I was humanizing the fish, but fish do actually have many genes that are duplicated compared to other organisms because of a teleospecific whole genome duplication event. And so, there was this result we had where when we knock out SRGAP2 in our fish, we don't show embryonic lethality, whereas in the mouse, there is embryonic lethality. And so we can characterize phenotypes in our fish that maybe you couldn't do in a mouse because the mouse would never grow up past a certain age. And so I think to that regard, I think it might be possible that there's potentially more genetic compensation occurring in the zebrafish because of these duplications that exist in their genome. And so perhaps phenotypes aren't as severe, but you can still understand molecular characteristics of development in these fish. And it's allowing us to kind of get over that hurdle of this embryonic lethality. So I think in that regard, I think the zebrafish is useful there.
Thank you.
Thanks. So the other kind of big discussion, hopefully this brings in all the models, but from a phenotype clinical side down or up, whichever direction you're looking at this. Eric and Anne and others maybe want to start us off, but basically I'm curious, hopefully, from an animal model side, what are the key things that would be LGS specific enough? I guess that's what I put it in, that you prioritize, given the idea that a model might not is definitely not going to capture everything you want to see because they're not patients, but maybe each of these models has an advantage to mimic a specific piece of LGS that we can then look for therapies or treatments or mechanisms in.
If I may, I think part of the reason Eric and I got into this push me, pull you conversation yesterday was that, I think it really, and we saw yesterday these great talks about very specific ways to interrogate the human network dysfunction, right? We actually had readouts that were not just EEGs, which we see in our patients, and not just the seizures that we see in patients, but really sophisticated ways to look at the network dysfunction. And so that's wonderful. And we've refined the phenotyping there, which is great. Not every single one of those is going to be recapitulated in every single model. There's still interesting models and it's really cool science, but how do we connect the two? And so Kelly's question, I think, was kind of getting at what I was thinking about. If we're thinking about taking these human features and then modeling some of them in animals, or in cell cultures or in organoids, and then developing drugs or using those as platforms to test drugs for LGS, what are the key core features that we are going to require? So when someone says, hey, here's a drug we want to pilot in the LGS population, which of those validities you mentioned are going to be met? It seems really simple, but I think there are gaps that we see when people come to us and say, hey, here's a drug you want to try for this condition. Well, what are the preclinical features of what models that have been met? And it's often that they're not quite aligning. So I think, the example you had of the serotonergic drugs really going from animal to human and actually having all of those validities met is really the kind of example we're looking for LGS.
Kelly?
So then it becomes what are those features, right? Is it from the EEG perspective? Because if LGS is an electroclinical diagnosis, right? That's what we're talking about. So it's the electrical part, right? Is slow spike wave GPFA at some point. So which, do you need both? Do you need one? I would say either would be sufficient, right? If you had a model that had GPFA or slow spike wave, it would tell you that the network is able to generate the type of activity that the speakers yesterday were talking about. It would be great to know, right, in a mouse or in a fish. And that's what I was kind of, that you would having not just, if there is this brainstem deep evolution to the cortex that happens in GPFA or in spasms, that you can see that in a fish because those structures are there in a fish, even though the cortex is not. And that might allow you to model those type of things. So is that the key? Is that all we really want is just the electrographic aspect of it? Is that enough? I don't know what you guys, Kelly, what do you think?
We'd all say no.
I guess, yeah, for me, you'd kind of like to see the clinical seizures as well. Some of our discussion yesterday was about, what we'd ideally like to do is prevent the evolution into Lennox-Gastaut, which requires a lot of work on the clinical side that we have to be able to predict who would be at risk of evolving into Lennox-Gastaut. But on the modeling side, it also means we need a model that we can see evolves from one thing into Lennox-Gastaut. That's part of the disease. And if all we're looking at is to make the seizures go away, that's no different than what we have right now. But to really impact the syndrome, I think we need something that recapitulates that evolution.
Yeah,
So-
I would love to. I'm sorry, who was talking?
Go ahead, Tracy.
Okay. Yeah, I think that I think that just given where the state of the science is right now, that any feature other than just seizures would be good, right? So if somebody is targeting like Ming Shan, right, or Megan, where there's targeting specific genes and they're looking early, that is a form of prevention. But for us at the LGS Foundation, we want to focus on the Electroclinic, the electrographic, features. We want to focus on slow spike and wave. We want to focus on GPFA. If you can find that in a model today and you can target that, or if you can target it in humans, I feel like that's one step forward, even if you just have one. And if you could start to dissect, let's say you do have an animal model with slow spike and wave, which we think Dr. Carney has, right? You could start to dissect out, where is that coming from? Not just what are the brain structures, but also, what are the cellular molecular mechanisms? And then you can also think about how that might correlate to humans, even if that's the only thing you see in an animal, because I think drug development in LGS is going to continue. We're going to continue to throw everything in the kitchen sink at LGS. There is a path to approval. For now, it's getting harder and harder to show superiority, but there is a path forward to approval. And thank God they're developing more treatments for us because we are desperate. We try everything and we need that help. But as we think about the science of the future, I don't want to fall into the hole that the infantile spasms community had where they said, you have to have all of these things. Even dissecting out tonic seizures are a huge problem in our community. And I would say maybe we don't want to target drugs just to tonic seizures, but they're a huge problem. They're one of the most difficult to control that I've heard from physicians. So if we could focus and try to solve that problem for the LGS community, if we could focus on any of these other features, that would be more precise than what we're doing now, which is simply literally dartboard. Let's just try this thing, see if it works. And if it passes through the it's not called the ad program anymore, the ESTP program, where it passed through the screens, then it's potentially a drug that can be tried in LGS. Or if it's passed through and have been approved in another treatment, it's potential to try in LGS and get orphan status for.
So Tracy, 2 points. One, I agree. The infantile spasm consortium approach that set strict criteria that a model had to have all of before they would like it, I think was ill-fated. But with LGS, and so this is more of a support issue, because I can imagine it more, even between the four of us here or any 4 or 5 labs that have different technical approaches, that you'd model, each of us can model a specific feature in, organoid, fish, mouse, whatever. And you can get all 4 or 5 features, maybe the clinicians would agree together are the features of LGS, but each of the systems might only have one, a robust one, undoubtedly, because if you want to think about drug screening to be true, and Allison kind of hinted at this, the variability of your phenotype is a critical feature if you're going to find a drug. So if you have a model system and the baseline is all over the place, it's hard to see above that noise. But if the phenotype's really strong, and so our advantage, for example, which is belied by the way it's presented, usually by me, is that the Dravet zebrafish model had a seizure every minute. And that's what Jennifer is hinting at. And so a fish where hundreds of fish are having seizures a minute made it really easy for me to look for drugs that stop that. I have other fish in the EASY project, as Jenny's kind of hinted at, and others probably know, and Anne, that maybe have a seizure an hour. And so they might represent that disease, but they're going to be hard to drug screen for. And so I think, each of the models being optimized for the mice have tremendous potential for modeling, and interfering with different circuits to get specific behaviors that go with the physiology, like Minshan does and others do, especially as him and others, I'm sure, layer on in vivo recordings with that as well, in vivo physiology. And so you can mimic that really well. The organoids have very specific, manipulations that you could, like I said, put them together the way you want to get a feature you want. The fish have certain features that you can do very quickly in a week, that allow. And so my view is that, and I don't know how LGS wants to go, I guess that's the next discussion about funding and other things like that. But I can imagine if there was a couple therapies we wanted to test as a group, you wouldn't necessarily want to just pick one of us. I'd say pick 5 or 6 people with the same therapy, because I think that's the more efficient way to test a therapy. When you think, I think in gene therapy or specific classes of drugs that people already said, this class could work for LGS, it's a lot easier to get data from different pieces and to get one piece of really good data from one lab and one model. And that leads me to my second part, kind of Anne mentions. My experience with the FDA is also very unique in this. And the interesting thing I found, which I did not know was going to happen because the ASP program, which uses non-genetic models, was that the FDA actually, for an epigenetics case, every time we petitioned them for going to animals, from animals to people, they've never actually asked us to do mice or other things. They just looked at the efficacy data on our model, and then they worry about safety data and whether there's a need for that drug. And because they've understood from the beginning, maybe not with Dravet so much now, that there's not an animal model for LGS where we can test a drug and go to the FDA and say, hey, this is our LGS preclinical efficacy data, because there's not a model for the majority of these rare genetic diseases. But my experience is the FDA doesn't care as much as the epilepsy community cares about that. They want to see efficacy in a reasonable model that's, that has the features like you said, and they want to see safety and a reason why, like you said, why that community is willing to test that drug. And so I think what's delayed us, after I learned more about the process, is the FDA is actually more helpful and willing with the rare diseases to help us along to the clinic than I think a lot of people realize. And
I think that's a really great point, Scott. I think there's some other questions if you want to pull that out, but I mean, I think just going, thinking about where we're at with the foundation, I think that there's also this component of what we can learn from humans, and Kelly alluded to this too, but we can follow humans as they go from tuberous sclerosis to infantile spasms to LGS and try to figure out more about what that evolution looks like, and that can be EEG, that can be other features, right? And so kind of going back to the talks yesterday, so there's that component, and then you can sort of cross compare, right? If you're seeing like the work from Dr. Archer, if you're seeing very specific networks that are involved in LGS, right? And Dr. Galinas, I think, wants to look into this a little bit as well. Then how does that sort of transfer over when you're looking at this group of investigators across models, and how are there correlations? So I really love this idea of working across models as we look. And you're right. I mean, we do have 6 FDA approved drugs and no real mouse model for LGS that we've agreed upon, right? There are models out there. And I think we probably have more models than we know, because I don't know that people have necessarily looked, right? Usually we just would look to the first seizure and then stop, or look to the first abnormality and stop. So I think there's a lot of potential there. So did you want to see if there's other questions?
Yeah, actually, you perfectly set up a question by Heidi Grabenstatter from this, I think Heidi's from the CDKL5 Foundation. This is for Min Shan. She asked, did you follow the SCX-BP1 mice to determine whether the epileptiform activity evolves? If so, do you see spike wave discharges in wild types as well?
So we did follow this animal to an older age. It is true, the typical SWD I presented earlier actually are changing to a different morphology, right? So whether that represent anything more close to LGS, no, I cannot say for now, but I think it is true, they are evolving, they're not exactly the same. So the second part, the question for the wild type animal, I think it's true. We always see a very low frequency of SWD in the CBD7 Black 6 wild type animal. And they have exactly the same morphology we see in the mutant animal, just much, much lower frequency. So that is something I think the community has accept now, that the wild type animal do have a very low frequency of those events. So.
Great, thanks, Min-Shan.
Yep.
Brenda, I think I saw you here. You want to ask your question directly? You want me to read it? Putting you on the spot, sorry.
I'm very interested in the mice in particular, because they seem to have the best or the closest structure to what they were talking about yesterday, with a sort of a cortical, subcortical loop that you can create or look at and study.
Brenda, your sound is breaking up, Brenda.
Cortical, subcortical loop.
Right, so I'll read it now. Yeah, it's a little bit broken. In the chat, you asked whether mice are the only system that we have for that, or the best system for studying cortex and subcortical networks that we talked about yesterday. Or I would say the mice maybe are you asking if the mice are the best available animal model in a laboratory, or model period? Because there might be other places to model that in a laboratory.
Yeah. Sorry. You can't hear me, but
now you're muted.
Never mind.
Now we can hear you. Actually, now we can hear you.
Now we can hear you fine. So you're good.
Hey, go back on, Brenda. You were good.
While Brenda's deciding she'll jump back in, Megan has a question for Allison.
Allison, you're finding in your CDKL5 organoid model, I believe that's what it was, where you found that initial spike in activity and then a decay. Was that associated also with any kind of changes in, I guess, like composition of cells in the organoid versus the wild type, or like a developmental kind of defect in your organoid that could be
we think it's a developmental defect and not related to cell type, although we haven't investigated that carefully if the same populations are represented in the two organoids. We are doing this kind of single cell analysis right now. It might be that even like small decreases in some certain cell populations might reflect that. We'll see. We don't know. We think it's a developmental delay.
Okay, I see.
I guess proactively, because I see you both here, Jennifer Galinas and Kathy Chavon. Other features from the more complex electrophysiology that you guys use as markers in these types of things like LGS, you think we should be focusing on to model above others?
I was actually thinking about some of the network analyses that have been done on LGS. There are parallels with some types of focal seizures for which patients currently may get epilepsy surgery procedures done. So there's begun to be some, there's a pile of work looking at different seizure onset patterns in hippocampal epilepsy from both human and rodent standpoints. And those are the, sorry, cat. Those are the low voltage fast and the high voltage types of onsets. But there are tonic seizures that are studied with intracranial electrodes, and also we have some in microelectrodes. And there are insights that can be gained from studying those, because my sense is that there's different pathways to get to the same physiologic or pathophysiologic seizure onset sequences, and that you might be able to address at least some of the seizure types in LGS by studying the analogous ones in focal epilepsy. And the work of Jean Paz, for example, is an interesting bridge to looking at the relationship between thalamic seizure triggers and what look like focal epilepsies if you're studying intracranial electrodes. So this is another path that can be looked at. So that's my thought.
For me, the electrographic findings that I would absolutely love to see in a model is number one, kind of broadly, a disruption of the normal background. So in patients, we always see that patients with LGS typically have slowing of their backgrounds. Like the typical rhythms that we see are slow, and we extrapolate that to correspond to the intellectual and cognitive and developmental issues. But I think just at a basic level, it would be really interesting to see if our models could recapitulate that. Do we see abnormalities, in kind of the background electrographic and network functions in our models? That's the first thing. And then the next 2 points have already been discussed, but for me, they're the highest target value for LGS is the generalized paroxysmal fast activity, and something that looks similar to a generalized spike wave discharge or the slow spike in wave. I think those are those three things is what I would be looking for electrographically.
Thanks. Jurian?
Oh, I'm sorry, I missed the last bit.
I just wanted to, I'm sorry to call you out unexpectedly, trying to get a sense of what are the most prominent electrographic features that, if we could model them, you'd want us to focus on.
I think the generalized paroxysmal fast is the most important, and the tonic seizures are occurring in other forms of epilepsy that do not quite meet the clinical criteria for LGS. And the slow spike in wave seems to be a pattern that may be present in young people, but only in half or so of adults. So that may be whether that pattern is related to the pathogenic process would be a very interesting question to ask. Like, if we were to be able to suppress these spikes, which may respond sooner to medication or medical interventions than the seizures themselves, would we be able to slow down the process? But the generalized paroxysmal fast activity, I think, is more specific to LGS, and that has some good overlap with other rapid patterns of generalization, including the tonic seizure. So I think that's a good target. And then Dr. Archer already, has been trying to isolate these spells on the EEG fMRI, and I think we can try and find patients with very similar presentations with and without generalized paroxysmal fast activity, and then see if we can map these networks specifically to this pattern, for example, with lesion network mapping.
Thanks. What before I get on to the other mass question, on the flip side, what are the specific, I guess, most prominent comorbidities? Because sometimes those are a little easier to model across animal models, behaviors or other features. I mean, both Minshan and Megan have the ability to kind of model a variety of early developmental behaviors, basically, at various degrees. And so it seems there's obviously a nice consensus on the electrographic features. My understanding has always been, there's a mixed bag on the genetic predispositions because there's many possibilities. And so it's on a single gene, and that's been studied for a long time. But is there a kind of a consensus on the comorbidity side as well?
Well, I think just one thing that would be consensus would be some level of broad cognitive dysfunction, intellectual disability, right? That is a core feature and would be needed. If you had a mouse that was a LGS mouse, but did perfectly fine in every test you gave it to it, right? That wouldn't be LGS. You might use it, going back to your points, right? You might use it if it had slow spike wave, but it wouldn't be LGS if it didn't have the cognitive dysfunction. So I think some level of diffuse network cognitive dysfunction is required as a comorbidity fundamental component.
I guess, I would agree, but I'd also say, it required, if you're going to develop a drug and if you're going to claim that drug can have developmental improvement, and then propose a trial where that's the outcome, right? I think we're all aware that not every genetic model of epilepsy produces seizures or electrophysiological abnormality. And so if you get one that does, and you can find a drug that can suppress seizures in that model, then if it doesn't necessarily happen in a model where you can look at development, like iPSC-derived neurons and developmental cognitive function, as Scott said at the very beginning, doesn't mean you throw the model away, doesn't mean it's a bad model. It's just only a model for that one particular aspect. And again, because we don't have a lot of precision therapies, maybe you do take what you can get in terms of the models producing really beautiful work and deep understanding about the science. And if there are some clinical features that are recapitulated, it's legitimate to say, well, we're going to look at response in humans to the human correlates of those features. And maybe your development becomes a secondary outcome, or vice versa. We've had proposals come our way for single case treatments where, a mouse has developmental improvement and somebody wants to use it to treat the epilepsy. I'm like, well, that is a nice idea, but maybe doesn't make sense. But maybe we could propose an experiment in the humans where we look at the development and have the seizures be the secondary outcome. I think we have to be clear and flexible at the same time.
That brings up a really good point, and there's actually a real need for neocortical epilepsy models and in general, and there's been a huge focus on hippocampal models for very good reasons, but I think also rodents just seize much more easily from the hippocampus, as do, I suppose, humans. But the big objection that comes up in study sections is there's a lot of heterogeneity in neocortical syndromes, and that's true, but it shouldn't stop us from studying neocortical models. So I've drifted into tumor-associated epilepsy because I was interested in neocortical seizures, and that was one way to approach it without getting caught in that little heterogeneous neocortical syndrome trap. And I don't really know how to get past that other than what I've done with, going into a different type of epilepsy. So, this is something I've racked my brain about for a while. And if anyone has suggestions, I mean, there have been efforts in that direction, like the tetanus toxin in models or the 4-AP injection. And there's big problems with both of those models. Or the Illumina gel, which is, another one that's also had a lot of problems. So, this is something that just needs a new effort, and then maybe some recognition that even with the heterogeneity that exists, we should still be able to do something there. Okay.
Thanks, Catherine. Yeah, I agree. I get that question a lot about modeling neocortical epilepsies, and specifically I said, what's the animal model for focal epilepsies? Mice and most animals, it generalizes so fast, it's almost an impossible question to address. The gel model goes back to David Prince in the 1960s, and it's still kind of discussed as one of our best options. So that really shows, no offense to David, but that really shows the lack of progress we've made. We're both the gel model was early 60s, the TTX infusions were early 70s, right? But yeah, I agree. I don't really have an insight on how to do it, but I definitely feel the need of more thought about better neocortical features and models. Brenda has another question. Do you think the age window is much smaller for reversing the developmental and cognitive dysfunction? At least in humans, that would be likely. So perhaps the same is true in animals. I don't really know. I don't know if anybody want to address that.
I mean, we know anecdotally from cases like my daughter and others, right? And John Archer talked about this yesterday with his cases where even in adults who have some cessation of their seizures or improvement, or they stop seizing, they do start to gain cognitive ground again, right? And so, but I think that it's not ever going to be, it's not all of a sudden my 28-year-old is going to start acting like a 28-year-old. There's been enough brain damage now that you're not going to be able to reroute the circuit completely back. But earlier on, we never study these cases, but there are these cases of children, and Elaine Worrell's on the call. She could probably speak this better than I can, where the kid has LGS and you get seizure control and their brain, goes back to quote unquote typical, and they don't have LGS anymore. I think these people leave our community. We don't see them because our work's so severe, but it's not unheard of to see that pattern. They've got everything. They've got the multiple seizure types. They've got the slow spike and wave, the GPFA. They've got cognitive, they're starting to delay in development. They're intractable. And then you get control through brain surgery or some other means. And now they're, sort of not living the LGS life. Not that they're perfect, but I don't know, Eric, Elaine, do you guys want to weigh in on that?
So, I think that what you're describing is something that's very rare, at least in my experience. Maybe I'm just a bad neurologist and haven't gotten anyone to do that. But it's a very, very phenomenal. And if it is, it's often someone who has a focal lesion that's causing diffuse, right? That focal lesion quickly corrupts the functionally the rest of the brain. You take it out early and then they do well. And I think that's a unique circumstance. I think these are the unknown questions, right? Of how much of this is reversible, how much of this is developmental, what is the role of seizures in impacting the brain? I mean, Tracy, you said your daughter has brain damage. What does that mean? Like, are neurons dead? Are synapses not functional? Like, what does that mean that she has brain damage? I mean, I don't know her MRI, but a lot of these kids, their MRIs don't change over years and years and years. So what is going on, right? I mean, I think there's still a lot to know. So Elaine, go ahead.
Yeah, I would agree with you. I think it's rare, but I think, we certainly do see it. And I would agree with you too, that I think it's patients who do have some type of focal abnormality. We've had a couple of kids that really kind of started looking like Lennox-Gastaut, and MRI can be really challenging early in life. And then you get an MRI at 2 or 3 and you're seeing a little bit of fuzziness and you think that there's something there. You go after that. I've had a couple of kids, just thinking back, and that's a couple of out of quite a large number that I've seen, where I really think that we've been able to kind of save them sort of at the nick of time and pull them back from, developmental problems, autism. I've had a couple of kids that really from after surgery, really lost their diagnosis of autism and really started progressing developmentally. So I think you can do that, but I think there is a critical window. And I agree fully with Tracy that if you've had 20 years of LGS, you're not going to get this back. I think you have a critical window that you probably need to jump in and act before, 3 to 4 or 3 to 5.
think intuitively the window is larger, right? Because the process of developing LGS seems to be protracted over years rather than like weeks or months, what we see in the spasms, let's say, right? It's not as acute. So I would think that the window is longer rather than shorter as compared to more acute epileptic encephalopathies.
I mean, this goes to just the kind of the general concept, right? What is reversible for all the neurodevelopmental disorders? With using Rett syndrome of, oh, it's not reversible, then Adrian Bird's genetic rescue experiment saying, oh, maybe this is reversible. So like what is reversible? And it's possible it's reversible in a mouse, and in a human, we're not going to see that, right? We're going to see that there'll be some improvements, but no reversibility. And Rett kids sees all the time. So there's an epilepsy component to that too. So, what are we, what is reversible? What are we expecting? And, I agree with Jurian. I think the window, it might be much longer than we expect for a lot of these conditions. But I just don't think we know, I think, and this is where, unless we have a non-human primate model, I'm not sure any of the existing models do a good job of the longer term developmental aspect of this.
Can you hear me? I don't know. Oh, good. Okay. I guess I worry a little bit that if you do a study like Dr. Shu does a study in his mouse model and there's no, it's an adult mouse and there's no improvement in some neurocognitive testing that he's doing, that might suggest that it's not going to work in a human, but it might mean that you really just have to do it at an earlier time point. And then when we transfer that concept to our patients, we can't do it in a 25-year-old with Lennox-Gastaut due to STXBP1 mutation. We need to do it in a 3-year-old. So I guess I'm just thinking that we just have to be careful about the outcome measures and in what age range, because it does seem as though some of these things I worry are not going to be reversible. But if you looked at the right group, then it might be.
I was just wondering, in a whole other literature, there's a lot of information about recovery from brain injury. And, there's the concern about doing hemispherectomies later because you can't have reorganization of function. Or as much reorganization of function. I wonder if we couldn't maybe look at that literature and get some ideas of what might be expected in terms of that recovery time course and the plasticity. And traditionally we usually think of onset of puberty as sort of a critical time when that plasticity decreases. And I wonder if that's the case, and if we might think about considering that role as well in looking at LGS.
Yeah, I agree with Anne. I mean, I to answer your question, Eric, no, the MRI doesn't change, but I think that, it's a wiring defect. That maybe if you looked more closely at the way things wire, or if you did functional neuroimaging, you'd see what Dr. Archer was presenting yesterday. And, we always learn as neuroscientists that cells that fire together, wire together, right? And so there's a certain point where you can no longer compensate for the road that this, self-perpetuating network of interictal, slow spike and wave, and then this GPFA has created. So, but I do think there's a critical window. It's probably longer. I think that, and I was one of them, the people that sort of wrote off my own daughter as, oh, she's never going to get any much better. Too much damage has been done. And so I think that trying things in older adults is right. I totally agree with you, Brenda, that we have to do our experiments, I think, in a clean population. But I think what we're seeing anecdotally, at least, is that you can, get some compensation for this. There's a compensatory mechanism we don't understand, but also that you can reverse some of it. And Anne, I love that idea of looking at that literature. I see some other questions coming in, but I did want to ask Jennifer Carney if she could just share briefly about the work she's doing on her KCNB1 mouse that we funded here at the foundation. And then I know we're getting close to time here. So Jennifer, do you want to just comment a little bit about that's relevant? Great.
Yeah. So, this is one of those models where we made it and we started doing EEG recording. And it was really remarkable because it was the most unusual EEG we've ever seen from a mouse. And in characterizing it with a pediatric neurologist to try to keep it, at least trying to look at the same parameters and the same systematic approach that would be used in humans, in mice. And what we found is that we do have slow spike and wave. And unlike what was mentioned earlier, that mice have this kind of background of slow spike and wave that's around 3 to 5 hertz. These are slower. These are around 1 to 2 hertz. And we can see they come out of wake and out of sleep. We can see trains that go on for 15 minutes particularly out of sleep, but we also see them in wakefulness. And then there's also paroxysmal fast activity. So EEG-wise, it does a pretty good job of approximating some of the EEG features that we all just were talking about wanting in a mouse model. And in discussions with Tracy over a couple of years, she convinced me that, maybe this is an LGS model. And in fact, some of the kids with KCNB1 variants have been diagnosed with LGS. So it seemed to fit. In terms of behaviors and other features, that's a harder question because we can't tell you if they have cognitive dysfunction because they have such profound behavioral dysfunction. They're extraordinarily hyperactive and have high impulsivity and aggression. And we really just can't get them to attend to any behavioral tasks that we ask them to do. So cognitive dysfunction is unclear, but certainly neurobehavioral issues are in play. So we published the mice, the initial characterization earlier this year, and we're happy to share them with anyone. If you want to get in there and kick the tires and take a look at them, we'd be happy to send them. We are trying to deposit them at the MMRC at UC Davis. It's COVID kind of put a wrinkle into that, so it's been taking a long time, but I'm happy to ship mice. We ship mice to people all the time, so that's easy to do as well.
Jennifer Glenis was asking the age of the mice when you see this. I think it was like P21.
Yeah, we've done it. We have done it as early as P21, and then we also have some adults, and there is some evolution over time. So you get more of the slow spike and wave as they get a little bit older. And then they do develop spontaneous generalized tonic-clonic seizures, but that's more like 4 to 6 months. But these EEG features are there early on. We haven't done much because of just the size of the mice. We haven't done earlier than P21, which is something that it would be really nice to have some pre-weanling EEG, but that's a little bit of a trickier thing. And maybe Jennifer would be able to do something like that, but with what we have, we're not able to do it yet.
Yeah, I would be happy to talk more about that because we've been recording in mice between 5 and 14 days of age, not chronically, but at least right now, but at least acutely for a few hours, so you can get a good sense of the interictal background. And so would be happy to discuss more with you about how we might be able to track the early development of those really fascinating interictal features.
Yeah, that would be great. And then I think there is background slowing as well. I don't think I mentioned that, but yeah, we're definitely happy to send them to anyone and see what happens.
Fantastic. I think that's great. Well, I think if there are any more lingering questions, go ahead and unmute now and you can ask them. But I think we're winding down here. We're almost at time. Anyone on the discussion any final questions here? Okay, well, with that, then I think we can break early. Thank you so much, Megan, Allison, and Mingshan for an excellent talk. They were just really spot on. I thought this discussion was excellent. Scott, thank you so much for pulling this amazing group together and for leading such a thoughtful discussion. And I hope you guys will all join us for the afternoon session, which starts at 2 p.m. Eastern time. So 2 p.m. on the West Coast for anyone that's here from another country, New York time, where we are going to talk about strategies for funding in the future for the foundation, but also other gaps in the field more broadly as we start to think about how we might move things forward in the LGS field as a whole. So hope you guys will join us for that. Eric Marsh will be leading that, and we'll have comments from Martina Bebin, from Yvonne Soltej, who is here, and also Ann Paderti is going to be stepping in for Jeff Nobles, who is cleaning up from the hurricane. Ming Shan, I think you won the award for most dedication today.